Hello, this is Dr. Kudakasseril. Today I am going to talk about one of the techniques used in amplification of the gene, namely polymerase chain reaction. This is one of the very important techniques used in biotechnology. Polymerase chain reaction, or PCR, was invented by Mullis. He got Nobel Prize for this invention. PCR is commonly used in laboratories to make millions or billions of copies of particular region of DNA. So here we are making millions of copies of the foreign gene, which is called gene cloning. Let us look at the requirements of PCR. We need a thermal cycler, which is a machine used for PCR, DNA to be amplified. In our case, it is the foreign gene. Then we need Taq polymerase. Taq polymerase is a DNA polymerase which is obtained from the bacterium Thermus aquaticus. This bacterium is found in hot springs. Therefore, the DNA polymerase of this bacterium can function at high temperature. We need 4 types of deoxyribonucleotides, or the nucleotides found in DNA. We need DNA primers. These primers are single-stranded DNA, around 20 nitrogen bases long, which are complementary to the 3' ends of the DNA. We use primers because Taq polymerase cannot initiate replication from the ends of the DNA. It can only extend a primer. We need 2 primers because the 2 strands of DNA go in opposite directions. One is 3' to 5', the other is 5' to 3'. These ingredients are assembled in a tube and put through repeated cycles of heating and cooling that allow the DNA to be amplified. Now let us look at the steps of PCR. PCR has 3 steps. First step is called denaturation, in which DNA is heated to 95 degrees centigrade to separate the 2 strands of DNA, or denature the DNA. In the second step, DNA is cooled to 55 degrees centigrade. That is when the 2 DNA primers that we have synthesized bind to the 3' ends of the DNA. This is called annealing. In the third step, which is called primer extension or chain elongation, we heat the DNA to 72 degrees centigrade, which is the optimum temperature of Taq polymerase. Taq polymerase binds to the 2 primers and extends the DNA in 2 directions, or in other words, replication occurs. So after replication, we get 2 copies of the original DNA. This is called one cycle of replication. This is repeated again and again till millions of copies of DNA are obtained. After 30 cycles of replication, which takes 90 minutes, we get 1 billion copies of DNA. PCR has many research and practical applications. It is routinely used in DNA cloning. That is, in genetic engineering, we clone the foreign gene before insertion into an organism, which is also called amplification of the gene. In the second application, we use PCR to find out the presence of a pathogen in the host. For example, in COVID-19 testing, we use PCR to find out the presence of the coronavirus in the host. Third, forensic analysis of DNA, or in other words, in DNA fingerprinting, the DNA present at the crime scene is so small in quantity, we have to amplify that DNA using PCR. Now let us look at RT-PCR, or reverse transcriptase PCR, which is the technique used in COVID-19 testing. Coronavirus, which causes COVID-19, is a retrovirus, or an RNA-containing virus. From the RNA of this virus, a complementary DNA is produced using reverse transcriptase. This DNA is then amplified using PCR. So PCR is a very important technique in biotechnology with a lot of applications. Thank you for listening to my video.
I have published some books for students appearing for the NEET exam. One is Handbook of Biology, Volume 1, which describes all the chapters in class 11, and Volume 2, which describes all the chapters in class 12. And also I have published a book, Biology in a Nutshell, which includes all the chapters. These books are available on Amazon. I am giving the link below.